Okay, HL, let's give these video notes a try. Let me know if this is helpful for you and I'll keep doing them. Let's do the SL review notes for D1.1, and that is DNA replication. So the point of DNA replication is to produce exact copies, right here. The point of DNA replication is to produce exact copies of DNA with identical base sequences. This is required for reproduction and for growth and tissue development in multicellular organisms. What does that mean? Well, that means that from time to time, your cells are going to need to reproduce. Now, we're not talking about human reproduction here, but what we're talking about is you have one cell and it needs to divide into two cells. That's reproduction that we're talking about. Also, very similar to that is growth and tissue replacement. Growth is just when you're physically growing, you need to make more cells. We should draw that. So when you are growing into this mature person that you are becoming, you are going to need to make more and more and more and more and more cells. And so you're going to need to copy your DNA so that all of those cells have that same DNA. And the same is true for tissue replacement. So if you get a cut, you're going to have to replace those cells with exact copies of the ones that were there before. And so you're going to need to copy your DNA in order to do that. DNA replication is semi-conservative. Let's see, semi-conservative. There's that word right there. Semi-conservative means that each new double-stranded DNA molecule contains one of the original base strands. So here we've got this picture where we've got our two dark blue DNA strands. And what's going to happen is these two are going to separate. And one is used to make a copy, a complementary copy. And the other one is also used to make a complementary copy. So what do we mean by complementary copy? If this dark blue one reads A, T, C, C, T, G, then the purple new one is going to read T, A, G, G, A, C. And vice versa. If our original strand here read A, T, C, C, T, G, that means that our dark blue strand read T, A, G, G, A, C, which means that the complementary strand reads A, T, C, C, T, G. Now you can see that these two strands here are exactly the same as these two strands here, and that's the point. The point behind using the complementary base pair rules is so that our new strands that are made are exact copies of the ones that were there before. And so we use one of the originals as a template. So each new strand, right here, each new strand is a complementary copy of an original, which allows for a high degree of accuracy in copying base sequences. So because of this complementary base pair rule, where A always pairs with T and G always pairs with C, you're able to keep the original sequences there. Now in SL, you were supposed to know about two specific enzymes. Those enzymes are helicase and DNA polymerase. Helicase is an enzyme. It unwinds and separates the DNA from each other by breaking the hydrogen bonds between the strands. So remember that between A and T, there are 2 hydrogen bonds, and G and C, there are 3 hydrogen bonds, and helicase unzips those. Now, DNA polymerase is our next one. The DNA polymerase enzyme, well, it's going to act on the DNA. It's a polymerase, which means it's going to build a long chain of something. So what DNA polymerase does, let's clear some space here. DNA polymerase reads each base and adds a complementary nucleotide to the growing strand. So if this blue strand read T, A, C, our complementary strand is going to have an A, a T, and a G put there by this DNA polymerase enzyme. This happens pretty fast, about 50 nucleotides per second. It's able to do that. But your set of DNA has more than 1 billion bases. So during mitosis in the S phase, when the DNA is replicated, so in the S phase, which is the part of mitosis when DNA is replicated, because S is for synthesis. When DNA is replicated, it would take about 8 hours to copy all billion, more than 1 billion bases that are in your particular genome. All right, so what can we do with DNA once we know how to copy it? Well, there's 2 things that we can do with DNA when we figure out how to copy it. And those 2 things are, we can now do a polymerase chain reaction, and we can do something called gel electrophoresis. Polymerase chain reaction takes a small amount of DNA and makes more copies of it. This is super helpful if you are a detective taking DNA samples from a crime scene. You have a very small amount of DNA when you get a DNA sample normally, and you need to make a ton of copies of it so that you can do different tests to it. So polymerase chain reaction, or PCR, will allow you to do that. Now, pause the video now and use the links that are on our daily slide to take notes on the role of primers, why we have to change temperature in PCR, why we have to use polymerase from a different species, and, well, I just kind of gave this one away, but when is PCR used? And then come back here and check your answers. Okay, I'm sure you went and did that. So let's see how we did. When is PCR used? When you have a small amount of DNA and you need more copies. All right, so how do you do it? Well, first, you got to heat up the DNA, which separates the strands. It denatures the DNA. Then you use a primer. A primer creates a small copy of a certain range of that DNA. You heat it up and you use, so the primers make these small bits of DNA there. And then after you have heated it up, you're going to make copies of those DNA strands. Now, we use a different kind of polymerase than our cells use. We use something called Taq polymerase. Taq polymerase comes from the species Thermophilus aquaticus, which is a bacteria that lives in very hot water. Why is this important? Because we have to heat up our sample quite a bit. And because we heat it up, we can't use our human DNA polymerase because that enzyme will just denature. This Thermophilus aquaticus normally lives in very hot springs, and so this enzyme will not denature when it's exposed to high temperatures, so it can make copies of the DNA. And as you saw in the video, after just taking 10, 20 turns of this, you'll have billions of copies of DNA. I might have my numbers wrong there off the top of my head, but you get the idea. Okay, now pause the video here and watch the couple of videos about gel electrophoresis. This separates molecules by size. When is this useful? And how DNA profiling works. All right, go ahead and pause, come back here and check your answers. Great, you're back. Okay, so what you got to do with gel electrophoresis is you get a small sample, you get a sample of DNA here, not a ton of it. I mean, there's a lot of molecules in there, but you add into it something called a restriction enzyme. This enzyme looks for very specific DNA sequences and cuts the DNA. So, for example, you might have a restriction enzyme that looks for ATTCTTA, and when it sees that in the DNA, it makes a cut. Those are my scissors. It makes a cut in the DNA right there. So what you end up with DNA, you end up with a bunch of DNA strands of different lengths, some longer, some super short, but there we go. Then you put that DNA in a gel. So the gel will have some little spaces here where you insert the DNA and you run some electricity from one end to the other here. Because DNA is charged, it will be attracted to the opposite pole. Now this gel is like, you ever look up really close at Velcro and see that it's a whole mess of strings? This gel kind of looks like that. And so as the DNA is pulled from side to side, the smaller bits are going to be able to get through all of those spaces. So smaller bits of DNA will move farther down this gel, and the bigger ones will get all tangled up in those gel fibers and not be able to go any farther down here. And so you add a little bit of dye to this. You can look at this under a camera and see how far things go. This is really helpful because if you have, let's see here, if we do paternity testing and you have the child here, and here you have two potential fathers, and here you put the mother of the child, because the father will share some DNA with the child, it will cut in the same spots and you'll get pieces of DNA that are the same size. And the same is true with crime scenes, but instead of fathers, you would have suspects and a victim. So you could compare suspect and victim DNA. Lastly, when we do a gel electrophoresis, one of the first things that we put in here is something called a ladder. That's a piece of DNA that we know exactly how big it's going to be cut up into. And so it'll literally make like a bunch of strips here in our gel. And remember, the longer ones don't go as far. And so this might tell us that if it goes this far, it's 100 kilobases long. That's 100,000 kilobases long, whereas this one might only be 1 kilobase long. And so we could actually tell how big the DNA is by where it sits. Okay, that's it. Rewatch what you need to and ask questions as you have them.